Dear friends, hello and welcome. Today I am going to deliver a lecture on forces involved in ligand receptor interactions and relationship between drug dose and drug effect. Now let us first try to have a look at the main objectives of today's deliberation. Number first is to give a general and thorough idea about the term receptor and ligand. Number second, explaining in detail the forces involved in ligand receptor interactions. Number third, to discuss in detail relationship between drug dose and drug effect. And finally, we shall be discussing in detail the graded and quantal drug response. Now, we will start with the introduction about the terms receptor and ligand. Receptors are macromolecules involved in chemical signaling between and within cells. They may be located on the cell surface, membrane, or within the cytoplasm. Activated receptors directly or indirectly regulate cellular biochemical processes, for example, ion conductance, protein phosphorylation, DNA transcription, enzymatic activity, molecules, for example, drugs, hormones, neurotransmitters that bind to a receptor are called ligands. In biochemistry and pharmacology, a ligand is a substance that forms a complex with a biomolecule to serve a biological purpose. In protein ligand binding, the ligand is usually a molecule which produces a signal by binding to a site on the target protein. The binding typically results in a change of conformation of the target protein. In DNA ligand binding studies, the ligand can be a small molecule, ion, or protein which binds to the DNA double helix. The relationship between ligand and binding partner is a function of charge, hydrophobicity, and molecular structure. The instance of binding occurs over an infinitesimal range of time and space, so the rate constant is usually a very small number. Binding occurs by intermolecular forces such as ionic bonds, hydrogen bonds, and van der Waals forces. The association of docking is actually reversible through dissociation. Measurably irreversible covalent bonding between a ligand and target molecule is atypical in biological systems. The etymology stems from ligare, which means to bind. Ligand binding to a receptor protein alters the chemical conformation by affecting the three-dimensional shape orientation. The conformation of a receptor protein composes the functional state. Ligands include substrates, inhibitors, activators, and neurotransmitters. The rate of binding is called affinity, and this measurement typifies a tendency or strength of the effect. Now, coming to the interactions involved in drug receptor interactions. Number first is the covalent bonds. These are strong, irreversible bonds with -40 to -110 kilocalories per mole stability. These are rarely seen in drug receptor interactions. More prevalent in drug enzyme and drug DNA interactions. Number second is the ionic interaction. This type of bond is weaker than the covalent bond. At the same time, it is one of the most prevalent bonds in drug receptor interactions. The drug molecule must have opposite charge compared to the ionized amino acids found in the receptor or enzyme. Number third is the ionic dipole and dipole-dipole interaction. Electronic dipole is formed when we have polar acid bond. In the polar acid bond, one of the pole will be partially positive and the other partially negative. These partially positive or negative charges might form an electrostatic bond with either partially charged atoms or ionized elements. Fourth is the hydrogen bonding. It can be considered as a subtype from dipole-dipole interactions, in which X can be nitrogen, oxygen, or fluorine, and Y can be atom with non-bonded pair of electrons such as nitrogen, oxygen, and sulfur. Stability of this bond is -1 to -7 kilocalories per mole. These are of two types: intramolecular hydrogen bonding, which occurs within the same molecule, and intermolecular hydrogen bonding occurs between the two nearby molecules. Number 6 is the charge transfer bonds. It occurs between an electron donor group in one molecule and an electron acceptor in another. Electron donors such as alkenes, alkynes, and aromatic ring bearing an electron donating group and atoms having pairs of non-bonded electrons such as oxygen, nitrogen, and sulfur. Electron acceptors such as aromatic ring bearing an electron withdrawing groups. Number 7 is the van der Waals forces. It occurs due to temporary non-symmetrical distribution of electron density. This will form temporary dipole that will interact with nearby dipole. Stability accounts for only -0.5 kilocalories per mole, which means that this type of bonds are much weaker than other bonds. The interaction of most ligands with their binding sites can be characterized in terms of a binding affinity. In general, high affinity ligand binding results from greater intermolecular force between the ligand and its receptor, while low affinity ligand binding involves less intermolecular force between the ligand and its receptor. In general, high affinity binding results in a higher degree of occupancy for the ligand at its receptor binding site than is the case for low affinity binding. High affinity binding of ligands to receptors is often physiologically important when some of the binding energy can be used to cause a conformational change in the receptor, resulting in altered behavior of an associated ion channel or enzyme. A ligand that can bind to a receptor, alter the function of the receptor, and trigger a physiological response is called an agonist for that receptor. Agonist binding to a receptor can be characterized both in terms of how much physiological response can be triggered and in terms of the concentration of the agonist that is required to produce the physiological response. High affinity ligand binding implies that a relatively low concentration of a ligand is adequate to maximally occupy a ligand binding site and trigger a physiological response. The lower the K level is, the more likely there will be a chemical reaction between the pending ion and the receptive antigen. Low affinity binding, that is higher K level, implies that a relatively high concentration of a ligand is required before the binding site is maximally occupied and the maximum physiological response to the ligand is achieved. In the example shown here, two different ligands bind to the same receptor binding site. Only one of the agonists shown can maximally stimulate the receptor and thus can be defined as a full agonist. An agonist that can only partially activate the physiological response is called a partial agonist. In this example, the concentration at which the full agonist, which is represented by the red curve, can half maximally activate the receptor is about 5 × 10⁻⁹ molar. Ligands that bind to a receptor but fail to activate the physiological response are receptor antagonists. In the example shown here, ligand binding curves are shown for two ligands with different binding affinities. Ligand binding is often characterized in terms of the concentration of ligand at which half of the receptor binding sites are occupied, known as the IC50, which is related to but different from the dissociation constant. The ligand illustrated by the red curve has a higher binding affinity and smaller Kd than the ligand illustrated by the green curve. If these two ligands were present at the same site, more of the higher affinity ligand would be bound to the available receptor binding sites. This is how carbon monoxide can compete with oxygen in binding to hemoglobin, resulting in carbon monoxide poisoning. Binding affinity data alone does not determine the overall potency of a drug. Potency is a result of the complex interplay of both the binding affinity and the ligand efficacy. Ligand efficacy refers to the ability of the ligand to produce a biological response upon binding to the target receptor and the quantitative magnitude of this response. This response may be as an agonist, antagonist, or inverse agonist depending on the physiological response produced. Now, let us discuss the next part of our today's lecture, that is drug dose and drug effect. Pharmacodynamics is the study of how drugs have effects on the body. The most common mechanism is by the interaction of the drug with tissue receptors located either in the cell membranes or in the intracellular fluid. The extent of receptor activation and the subsequent biological response is related to the concentration of the activating drug, that is the agonist. This relationship is described by the dose response curve, which plots the drug dose or concentration against its effect. This important pharmacodynamic relationship can be influenced by patient factors, for example, age, disease, and by the presence of other drugs that compete for binding at the same receptor, for example, receptor antagonists. Some drugs acting at the same receptor or tissue differ in the magnitude of the biological response that they can achieve, that is, their efficacy, and the amount of the drug required to achieve a response, that is, their potency. Drug receptors can be classified on the basis of their selective response to different drugs. Constant exposure of receptors or body systems to drugs sometimes leads to a reduced response, that is, desensitization. The dose-response relationship or exposure-response relationship describes the change in effect of an organism caused by different levels of exposure or doses to a stressor, usually a chemical, after a certain exposure time. This may apply to individuals. For example, a small amount has no significant effect, a large amount is fatal, or to populations, for example, how many people or organisms are affected at different levels of exposure. Studying dose response and developing dose response models is central to determining safe and hazardous levels and dosage for drugs, potential pollutants, and other substances to which humans or other organisms are exposed. These conclusions are often the basis for public policy. A dose response curve is a simple XY graph relating the magnitude of a stressor, for example, concentration of a pollutant, amount of a drug, temperature, intensity of radiation, to the response of the receptor, for example, organism under study. The response may be a physiological or biochemical response or even death, that is, mortality, and thus can be counts, for example, mortality rate, ordered descriptive categories, for example, severity of a lesion, or continuous measurements, for example, blood pressure. A number of effects or endpoints can be studied, often at different organizational levels, for example, population, whole animal, tissue, or cell. The measured dose is generally plotted on the x-axis and the response is plotted on the y-axis. Other dose units include moles per body weight, moles per animal, and for dermal exposure, moles per square centimeter. In some cases, it is a logarithm of the dose that is plotted on the x-axis, and in such cases, the curve is typically sigmoidal, with the steepest portion in the middle. Biologically based models using dose are preferred over the use of log because the latter can visually imply a threshold dose when in fact there is none. The first point along the graph where a response above 0 is reached is usually referred to a threshold dose. For most beneficial or recreational drugs, the desired effects are found at doses slightly greater than the threshold dose. At higher doses, undesired side effects appear and grow stronger as the dose increases. The more potent a particular substance is, the steeper this curve will be. In quantitative situations, the y-axis often is designated by percentages which refer to the percentage of exposed individuals registering a standard response. Such a curve is referred to as a quantal dose response curve, distinguishing it from a graded dose response curve where response is continuous. Now, let us discuss the last part of our today's lecture, that is graded and quantal drug response. To make rational therapeutic decisions, it is necessary to understand the fundamental concepts linking drug doses to concentrations to clinical responses. The concentration response relationships for drugs may be graded or quantal. A graded concentration response curve can be constructed for responses measured on a continuous scale, for example, heart rate. Graded concentration response curves relate the intensity of response to the size of the dose and hence are useful to characterize the actions of drugs. A quantal concentration response curve can be constructed for drugs that elicit an all or none response, for example, presence or absence of convulsions. For most drugs, the doses required to produce a specified quantal effect in a population are not normally distributed, so that the frequency distribution of responses plotted against a log dose is a Gaussian normal distribution curve. The percentage of the population requiring a particular dose to exhibit the effect can be determined from this curve. When these data are plotted as a cumulative frequency distribution, a sigmoidal dose response curve is generated. The equilibrium dissociation constant of the receptor drug complex, that is Kd, is the ratio of the rate constants for the reverse, that is K2, and forward, that is K1 reaction between the drug and receptor and the drug receptor complex. Kd is also the drug concentration at which the receptor occupancy is half of maximum. Drugs with a high Kd dissociate rapidly from receptors. Conversely, drugs with a low Kd dissociate slowly from receptors. These effects impact the rate at which the biological response ends. Potency refers to the concentration or dose of a drug required to produce 50% of the drug's maximal effect as depicted by a graded dose response curve. EC50 equals Kd when there is a linear relationship between occupancy and response. Often, signal amplification occurs between the receptor occupancy and response, which results in the EC50 for response being much less, that is, positioned to the left on the x-axis of the log dose response curve than Kd for receptor occupancy. Potency depends on both the affinity of a drug for its receptor and the efficiency with which drug-receptor interaction is coupled to response. The dose of drug required to produce an effect is inversely related to potency. In general, low potency is important only if it results in a need to administer the drug in large doses that are impractical. Quantal dose response curves provide information on the potency of drugs that is different from the information derived from graded dose response curves. In the quantal dose response relationships, the ED50 is the dose at which 50% of the individuals exhibit the specified quantal effect. The median inhibitory concentration or IC50 is the concentration of an antagonist that reduces a specified response to 50% of the maximal possible effect. That is all about today's lecture. Hope you have understood it very well. Thank you very much and have a good day.